Hi group, I hope you're having a good week so far. Today we're going to get into Unit 5, which is antigens and types of immunity. As always, we're going to go ahead and start with a few terms. So the first term I want to talk about is antigen. An antigen, of course, is a protein marker on the surface of a cell that identifies that cell as either self or non-self. Antigens are also loosely called immunogens, okay? And they are capable of provoking an immune response. Then we have antigenic determinants. These are structures or molecular components on the membrane of a cell that are recognized as foreign. Antigenic determinants are also known as combining sites or epitopes. Okay? The immune system recognizes these as foreign. And then we have hapten. Hapten is a partial antigen. Okay? It reacts with antibodies but cannot induce the formation of antibodies, is antigenic and can elicit an immune response but won't induce the formation of antibodies. A hapten can attach itself to a larger molecule and act like an antigen. Some examples of haptens are urushiol, which is a toxin from poison ivy, and hydralazine, which is a blood pressure lowering drug. Those are two examples of haptens. Now, there's a couple different types of antigens that I'm going to discuss throughout this unit. We have autoantigens, which are self-antigens, and they trigger autoantibodies. We have alloantigens, which is a foreign antigen from the same species. So some examples of that would be blood group antigens from donor cells. And then we have heteroantigens, which are plant, microorganism, or chemical antigens. These are antigens that do not originate from one's own body. And here what I've done is I've provided a list of things that make good antigens. And what I mean is that they meet all the criteria that makes something an antigens. By good, I don't necessarily mean they're good for us. I mean that these antigens are stable, have a large molecular weight, they can trigger an immune response, and they are complex. If you have a structure that has all of these properties, then these are antigens, and these are good antigens in themselves. We've got bacterial antigens, and those would be things like antigens on capsules, cell wall antigens, flagella antigens, and fimbriae antigens. And then we have toxins. These are poisonous substances of plant or animal origin. We've got viruses. Viruses make good antigens. And then toxoids. This is basically a toxin that has been treated so it's no longer toxic, but it can still produce antibodies. One example of that would be vaccines. So we have a list that discusses our good antigens, and then we have some examples of poor antigens, or things that make poor antigens. We have carbohydrates, and they're really too small to be antigenic, or they're too small to be good antigens. We have lipids that are poor by themselves, but if they can actually be linked to a protein, they function more antigenically. And then we have nucleic acids. All of these are examples of poor antigens. Let's go ahead and talk about the cellular antigens of clinical importance. These are antigens that are clinically important to humans. We have the human leukocyte antigens, also known as the HLAs. These are very important in transplants. HLA antigens include the histocompatibility antigens or the tissue-compatible antigens, and they are present on all nucleated cells. So like I said, they are present on nucleated cells, not just leukocytes, but they are both found on tissue cells and leukocytes. These human leukocyte antigens are part of the major histocompatibility complex that mediates interactions of white cells and other cells. The major histocompatibility complexes determine compatibility in organ transplants. Other cellular antigens of clinical importance would be autoantigens. Autoantigens are self-antigens that produce autoantibodies. And then we have blood group antigens. These have properties that make them good antigens. And also we have microbial antigens. Microbial antigens are antigens on bacteria, viruses, and then toxins like exotoxins or toxoids. Let's go ahead and discuss what properties actually make up an antigen. So we're going to talk about the physical and chemical nature of antigens here on this slide. Foreignness. The degree the immune system recognizes the antigen as self or non-self. So something that is recognized as being more foreign is something that is probably going to be more antigenic. Degradability. An antigen must not degrade too fast. It must be present long enough to trigger an immune response, or it won't be antigenic. Molecular weight. We want something with a high molecular weight of greater than 10,000 Daltons. We want them to be large, complex structures for them to be good antigens. Structural stability. If it's too weak, it's not going to make a good antigen. And then complexity. The more complex the antigen is, the more effective it's going to be in eliciting an immune response. And then finally, an antigen is usually a protein in nature or a large polysaccharide structure. Let's go ahead and ask ourselves, what is the role of the antigen, or what is the criteria which an antigen must fulfill? First of all, it must induce the formation of antibodies or immune lymphocytes, and it must react specifically with the antibody that it produced. So now is probably a good time to talk about our types of immunity. The first type of immunity I want to discuss is natural resistance, and this is also called non-susceptibility. This is absolute protection against certain diseases. For example, man is not susceptible to certain animal diseases. Then we have natural immunity. This is immunity that you were born with and antibodies that are present without external stimulus. We've got natural immunity. This again is that you're born with, and this is affected by heritage or race. Then we have two types of acquired immunity. We have active and passive acquired immunity. Active immunity builds up and lasts for years, and that is from having the vaccine or having a disease. Passive acquired immunity is developed immediately, but it's only temporarily. This is the type of immunity where you have antibodies that go from mom to baby, or if you've been given an injection of gamma globulin. We have cell-mediated immunity, which is the production of sensitized T cells. And then we have humoral immunity, which is B cells that synthesize antibody. So those are the different types of immunity. On this slide, what I've done is I have outlined the differences between active and passive acquired immunity. And you can see there that the source of active immunity is self. In other words, you've been vaccinated or you've had the disease. In passive acquired immunity, the source is another human or animal. That's the source of your antibody production. So it may be produced by being given an anti-sera or transferred from mom to baby. The effectiveness of active acquired immunity is quite high, and the effectiveness of passive acquired immunity is moderate to low. The method of acquisition for active acquired immunity, like I mentioned, is that you've had the disease or you've been vaccinated. The method of acquisition for passive acquired immunity is that you've had antibodies go from mom to baby, you have antibodies in breast milk, or you've been given an injection of gamma globulin. The time to develop for active acquired immunity takes a few days, up to 15 days or so, whereas passive acquired immunity works immediately. The duration of active acquired immunity is something that lasts for years, so the duration is very long, whereas passive acquired immunity, the duration is quite short. You're only protected for weeks to months. And then finally, the use of active acquired immunity is prophylactic. Okay, so the agent contributes to the prevention of the disease, whereas in passive acquired immunity, the use is therapeutic or prophylactic. So therapeutic is an example of when you've been given gamma globulin after you've had an exposure to hepatitis or something like that. And then here I've outlined the differences between humoral and cell-mediated immunity. Okay. In the humoral immunity, the mechanism is antibody-mediated, while in cell-mediated immunity, the mechanism is cell-mediated. Okay, so humoral immunity originates from the bone marrow, and cell-mediated immunity arises from the thymus. Okay, the cell type in humoral immunity is B cells, and the cell type in cell-mediated immunity is the T cells, the T lymphocytes. The mode of action or how these work, the humoral immunity, we have antibodies in serum where the B cells stimulate immunoglobulin production. And in cell-mediated immunity, the mode of action is cell-to-cell contact or soluble products from cells. And those soluble products would be things like interleukins or other cytokines. Okay? And then the purpose of humoral immunity is the defense of some bacterial and viral infections, while cell-mediated immunity covers a few more things like viral, fungal, intracellular bacteria, tumor antigens, and graft rejections. So the ability to form an antibody depends on a number of things, including the nature of the antigen. So what are the physical and chemical properties of the antigen? Also, the ability to form antibodies depends on the dosage or mode of administration. How is the host exposed to the antigen? Was it an injection, close contact, vaccine, or placental transfer? And then finally, the last thing that we look at for a person's ability to form antibodies depends on characteristics of the host. Things like the host's age, disease state, nutritional or hormonal conditions. So those are all things that we have to consider when antibodies are possibly being formed. Okay, so let me give you a few examples of active and passive acquired immunity and see if you can tell the difference. So if you had an administration of hepatitis vaccine, would that be active or passive? That's right, that would be active. If you're recovering from viral pneumonia, would that be active or passive immunity? That would be active immunity because you recovered from the disease. You actually had the disease. If you've been given an injection of gamma globulin, is this active or passive? This is passive because gamma globulin or anti-sera is an antibody injection. Recovery from measles or rabies vaccination, those are both examples of active acquired immunity. And then let's see here, prophylactic serum from hepatitis, that's an example of passive acquired immunity. And then finally, if you have an elevated titer of anti-salmonella H antibody, that would be active immunity, because if you're forming the antibody, it means you've had the disease or you've been vaccinated. Antibody response is broken down into two categories, the primary response and the secondary response. The secondary response can happen due to a second exposure or booster shot. The booster shot is known as the prophylactic, in other words, it contributes to preventing the disease. Immunization, a nonspecific anamnestic reaction occurs when there is a rise in antibody titer due to some other stimuli. Okay? There are 4 phases in each response. One is called the lag phase, where there is no detectable antibody in serum. Okay? The second phase is called the log phase, where the antibody titer starts to rise logarithmically. And then we have the plateau stage, where the titer stabilizes. And then finally we have the decline stage, where the antibody is catabolized or destroyed during metabolism. Okay, so there's 2 categories of antibody response, the primary response and the secondary response. Okay, a primary response, usually it's the IgM, it is the IgM antibody that is first exposed to an antigen that responds. And then in a secondary antibody response, we have IgG, an antibody that is made upon a subsequent exposure to an antigen. Okay? In a primary antibody response, it's IgM that is the first immunoglobulin to respond to antigenic stimuli. The response occurs in the acute phase of the disease. The primary antibody response from acute exposure to an antigen depends a lot on the individual and the specific antigen exposed to. The detectable antibody usually occurs at about 5 to 15 days. There's a very fast lag phase where the latent period lasts only a few hours to days. It is actually slower than a secondary response. The log phase, you will see a sharp rise in IgM antibody. The plateau stage, the IgM antibody starts to stabilize, that titer stabilizes. And then in the decline, there's a sharp decline of IgM in this stage. A second stimulation by the same antigen causes a secondary antibody response. This results in higher antibody titers. In a secondary antibody response, IgG is the predominant antibody formed to secondary stimuli. T memory cells stimulate the antibody production. This response occurs in the convalescent stage of the disease. There are some IgM antibodies that may be present in the secondary antibody response, but those titers are very low. The lag phase is slow with a rise in antibody titer at about 1 to 3 days. Okay, and then the log phase, there's a fast rise in the log phase. In the plateau phase, the IgG antibody remains high longer than in a primary response. And then there's a gradual decline of antibody titer in the secondary antibody response. So as you can see here in this chart, the primary antibody response takes longer for the lag to happen than in a secondary response. Both primary and secondary response show a sharp or rapid rise in the log phase. In the secondary response, the plateau remains higher longer than in the primary response. And in the primary response, the decline is sharp, but in the secondary response, the decline is gradual. So in a primary response, the time after antigen exposure is usually about 5 to 15 days, while in a secondary response, the time after antigen exposure until antibody detection is about 2 to 4 days, so it happens a lot quicker. The major antibody detected in a primary response is IgM, while in a secondary response, it's IgG. The amount of antibody in the titer in serum for a primary response is low, and in a secondary response, it's high. And then finally, the duration of the titer in a primary response, the duration is short, and the duration of the antibody in titer in a secondary response is very long. So I hope you learned a few things today about antigens and learned a few things about different types of immunity. So we will see you soon for Unit 6.